Hey guys, in this video I'm going to be discussing T cells, and T cells are very similar to B cells, so if you haven't seen my video on B cells yet, I do strongly encourage you to watch that first. But what we will see is many parallels between the growth and development of T cells and their role that they play in assisting B cells in your immune system. And so at the high level, just like B cells, what will happen early on in this process is that you'll have a stem cell in your bone marrow, and this stem cell, if it's going to mature into a T cell, will migrate to your thymus gland. And your thymus gland starts with a T, which is the whole reason why we call these your T cells. And the very cool thing that happens with T cells, much like with B cells, is that we have a parent T cell here, and it turns into unique daughter T cells. And each one of these daughter T cells will have its own kind of antibody on it. So if I try to make these look like different receptors, what we can see is that these are unique daughter cells with their own T cell receptor. And T cell receptor is this component on your T cell that will bind to epitopes. So this thing will bind to an epitope on a pathogen, hopefully. And the hope is that if we had some kind of bacteria floating around in your blood, like E. coli, that one of your daughter T cells would have an affinity for this E. coli, and it would be able to generate an immune response to kill the pathogens before they kill you. And that is the hope. Now, the problem here is that the process by which your parent T cell is generating these unique daughter cells is entirely a randomized process. And what that means, what I'm trying to say by randomized, is that these T cell receptors that get generated on each one of the daughter T cells can essentially be anything, because the DNA is being shuffled randomly during this division stage of the T cell life. And the problem with that is what happens if we generate some T cell receptors that have affinity for proteins such as hemoglobin. And hemoglobin is a protein that is very abundant in your red blood cells. Its job is to hold oxygen and carbon dioxide and release them when appropriate so that your cells can do cellular respiration. And hemoglobin is definitely a protein that is what I would call a good guy. And by good guy, I mean that we definitely don't want to have an immune response to hemoglobin. Otherwise, we're going to waste a lot of energy and time dealing with proteins that we actually don't want to harm because they're part of our own immune system. And so the way your immune system overcomes this problem is that it will send your T cells to the thymus gland, and within the thymus gland is where you'll have a lot of endogenous, I call it endogenous proteins present, like hemoglobin or insulin or other kinds of your good guy proteins that you expect to find in the blood. And that is where your T cells will essentially learn. So in the thymus, T cells learn who is good. And the way it does this is a little Spartan and barbaric. If a T cell has affinity for hemoglobin or insulin or another good guy protein, it is killed, because the only kinds of T cells you want to have floating around your immune system or your body, you only want to have T cells that are going to be binding to bad guys and generating immune response when they see bad proteins like viruses, not the good guys. So within the thymus, all of the bad T cells will get killed, And the remaining T cells will be T cells that have affinity for essentially everything other than your good guy proteins, which is the bad guys. And so by the time your T cell has been taught what good and bad is, it knows what bad is now because all of its buddies who thought good was bad are dead. They move on to lymph nodes. And this right here, this green region right here is a lymph node. Lymph node. An important thing that's going on within your lymph node is that you have a lot of T cells and B cells very close together. So I'll say I have a T cell here, and we'll have a B cell right here. And in my first video when I was talking about B cells, I omitted a rather important piece of information, which is in order for B cell to become fully activated, it needs to be encouraged by a T cell because B cells can ramp up production of antibodies very quickly, and this can be very bad if the antibodies that B cell is creating have an affinity for good guy proteins, because the last thing we want is an autoimmune response. And this would be an example of like B cells antibody latching on to insulin, which is another protein that's heavily used by your body in order to import glucose into your cells. You really don't want to have autoimmune responses damaging your body. So in this case, to avoid your autoimmune response, your B cells will need to be encouraged by a T cell to actually produce a antibody after they have been bound by that antibody as well. So the story that will happen here, so if we had a B cell here and its B cell receptor was bound onto, I'll try to draw something decent. If we had this B cell here and it was bound onto, we'll say that's a virus, it won't yet start generating antibodies for this virus until a T cell comes along and binds to this B cell to say, hey, this is something that I've been activated by this same virus, and you should also start producing antibodies for this virus. So we have kind of a two-stage verification for cranking out antibodies. And this is so that we can be extra sure that when your B cells start scaling up production of an antibody, we're very sure that the virus or whatever pathogen that has bound onto the B cell's receptor, the BCR, which is essentially an antibody. So this is something I also forgot to mention in the last video. The B cell receptor is an antibody, and if that antibody has affinity for something, we're going to make a lot more of those antibodies for your immune system. So if this is the case, your B cell receptor has an affinity, and a T cell has told that B cell, yes, this is a very bad guy that we do want to make antibodies for because I too have an affinity for this same bad guy, and this happens within your lymph nodes. Now we can start making antibodies to start fighting off this infection. So this is statistically a very good way to do things, because what will need to happen if we consider people who have autoimmune responses, so like worst case scenario is that you have a B cell react to insulin or some other protein that is not bad. It's something that we commonly would find in blood, and another T cell would react to insulin. Now we have a problem. But this "and" thing here is essentially a multiplicative thing. So statistically, if you want to get math nerdy here, we can kind of think of the likelihood that a B cell would make it out of the bone marrow and have an affinity for insulin is, let's say it's 0.01, so 1% of the time, let's say the likelihood that we would have a T cell also reacts to bone marrow would be another very low probability. So when we multiply two very small numbers together, we get an even smaller number. So this would be 1 times 10 to the minus 4. And these are made up numbers I'm just using here, but this is just to kind of hit home why it's very important for this two-stage verification for your body to have a good response to valid targets. And so that's going to wrap things up for this general introduction to T cells and the role in your immune system. In next video, I can talk more about CD4 and CD8 receptors. But I hope you guys find this useful, and thanks for watching.